A landmark Nature study reveals three distinct neuronal populations in the human brain that separately encode content, context, and their combination. The subjects were 16 epilepsy patients with 3,009 single neurons recorded across memory-related regions like the amygdala, hippocampus, and parahippocampal cortex. In a scene-dependent image comparison task, the brain had to recognize what an image was and in which context it appeared. Three neural subtypes emerged: content neurons that respond to the image itself, context neurons that track the current task or rule, and conjunctive neurons that bind content with context. Content neurons were concentrated in the entorhinal and parahippocampal regions, while context and conjunctive neurons enriched in hippocampus and amygdala. This work validates at the human single-cell level that content and background do not simply co-occur in one neuron, but travel along parallel streams that converge at the hippocampus. The fusion of content and context is carried out by a small set of dedicated conjunctive units. In the study, conjunctive neurons formed about 2.3% of all recorded neurons, but their presence was statistically significant. There was notable temporal coordination. Some background neurons and content neurons fired within tens of milliseconds, suggesting a causal orchestration. These findings tie together earlier debates about context-sensitive hippocampal maps and concept neurons in humans. From an engineering view, the brain effectively uses a content background decomposition to support memory and recall. The authors described the brain as a living page with content pages and distinct background frames. Neuroscience details add depth. Content neurons show stable responses across context, echoing the idea of stable concepts. Background neurons encode task context, such as time, place, speaker, or instructions, forming an abstract scene coordinate system. Conjunctive neurons bridge content and background to form memory entries tied to specific events. The study notes fast, millisecond-scale co-firing between background and content neurons as a hint of causal sequencing. The narrative connects to broader literature on time cells, grid cells, and concept cells in human memory. From a cognitive science lens, the brain's memory is not a monolith, but a set of interacting layers and modules. Now, what can we learn for AI memory systems? Three engineering takeaways. First, move from dense, entangled representations to decoupled content and background pathways. Second, introduce an explicit combining and auditing module to assess content background compatibility before generation. Third, harness few-shot learning by stabilizing background knowledge and inserting new content into existing frames. This could be achieved with frozen background subnetworks and fast updating content subparts, akin to adapters but semantically distinct. Fourth, tackle long context memory by maintaining explicit background and content caches rather than letting a single vector space dilute early context. A practical retrieval scheme would index memories by content background sparse associations, not just position in text. The authors argue that AI will move from a flashy oil painting model to a modular, writable memory notebook. The implications extend beyond nostalgia for human memory to real-world AI systems in education, robotics, and research assistance. In a word, the brain offers a three-part system: content, background, and the conjunction that stitches them together. From this nature work, one can envision a class of brain-inspired AI architectures that separate facts from context by design. For researchers, future papers may map these elements to hippocampal time cells, entorhinal context cues, and conjunctive memory units. Now consider a class project. Build a three-stream transformer with dedicated keys for content and context. In software terms, think of a content adapter and a context adapter combined with a mediating synthesis module. For long narratives or exams, maintain a background scaffold while inserting new facts as content slips into the scaffold. Experimentally, you could test whether updating content affects background representations or whether background templates stay stable. As a closing thought, the study offers a practical metaphor. The brain is a living notebook with pages and binder rings. The three lines of evidence, content, background, and conjunction, point toward a path where AI gains robust, auditable memory. In subsequent work, researchers may relate these findings to time cells, grid cells, and concept cells in the human hippocampus and surrounding cortex. The Nature paper thus serves as a rare bridge from single-cell neuroscience to scalable AI architectures. If you want to dive deeper, a longer reader could be offered sections comparing this work with the memory literature in rodents and humans. A future class sketch could visualize a brain-inspired architecture cartoon with content, background, conjunctive modules, and external memory. Finally, the video emphasizes how decoupled representations and a dedicated auditing mechanism may reduce AI hallucinations.